All right. Hey, everyone listening in. I am Stephen Fleming. I run Traverse Science, which is a consulting company in the feed, food, and beverage industry. We help private companies, nonprofits, other groups, those who make products in this space, understand science, and do the research. And what you're about to listen to is a conversation with myself and Dr. Kelly Swanson, who is one of our scientific advisors on postbiotics. If you're not sure what postbiotics are, you should definitely tune in if you care at all about gut health. Actually, health of, in a range of senses, but the microbiome, if you've heard of a prebiotic or a probiotic or a symbiotic, I think this will be of interest to you. Um, and and so Kelly Swanson, if you're not familiar with him, definitely look him up. Just search Kelly Swanson, UIUC. He is the Kraft Heinz Company endowed professor in human nutrition. He's a professor in the Department of Animal Sciences as well as the Division of Nutritional Sciences at University of Illinois at Urbana-Champaign. A lot of words there. So you could look up a lot of his publications. He's very active. But I'd rather tell you things that you can't figure out from just looking him up or going on ResearchGate or Scopus or wherever, which is I got connected with Kelly. Well, first off, my business partner, who I co-founded this company with, Ryan Dilger, Ryan and Kelly shared lab space. And Kelly was trained by Dr. George Fahey, which is, if you don't know who he is a very big name in fiber. That's a conversation for another time. But anyways, they shared lab space, Ryan and Kelly did. And just by nature of me being in Ryan's lab, I would, I was about to say I would interact with Kelly, but I didn't actually. I think in 5 years of grad school, I said, I never said more than hi. And he never said more than hi to me either. Nothing against him. We would just see each other in the hallway. Hey, how you doing? Cool. Move on. We occupied totally different spaces, like scientifically and just professionally and personally. And so it's ironic now, or maybe not ironic, but to have him as one of our advisors and be able to have such a great conversation with him. What you can't get from looking him up is he is so easy to talk to. He is so knowledgeable. I think everyone in the pet food industry and in companion animal nutrition pretty much know who Kelly is. But in the relatively short time I've known him, he has been so generous with his time. He's one of the best emailers I've ever communicated with. So very speedy and detailed. And he's never made me feel inadequate for not knowing something, right? He's been really great at educating and walking me through things. There are those great professors that you just kind of feel intimidated by. And not to say he isn't intimidating, but he's very welcoming and approachable. And those are things that I think you won't be, maybe you can get from this conversation we have, but those are things that you won't be able to get from looking him up. So anyways, I'm rambling on here. We talk about postbiotics, which is a relatively new term. These things have been sold for a while. And, in this conversation, disclaimer, this conversation is 3 hours long. So listen to it in chunks if you wish. We're going to introduce what postbiotics are. Why do we really need a new term for them? Or is that actually helpful? How to measure them? What are they? Why are they important in the pet food industry or really the broader industry? And then we kind of ramble and we talk about publishing and biomarkers, as well as statistics and planning projects, really a host of things. There's more details in the description of this episode. So I'm glad to have you here. I guess my marketing team would be upset if I didn't say smash that subscribe button and hit like for more content. Ugh, feels awful to say. Anyways, this is put together at no, there are no ads. This is totally free to you. This is just us putting out marketing content, wanting to educate others. So we hope you enjoy. And enjoy.
Got to get the makeup guy in here. So.
Oh my gosh, yeah. get the lighting right.
Yeah.
Someone touch him up. The hair's not perfect.
Get a shot of Mountain Dew here before we get going. So.
Oh, I love that. Is that your drink, Mountain Dew?
Yeah, well, Diet Mountain Dew. I added up the calories years ago, and I thought, well, I better stop. Stop
I did.
having the real
Oh,
thing, but it's still not the best thing in the morning. But
I love that. That's so unique. Some people, it's like coffee, tea debate, and you just come in, you're like, no, 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 no.
yeah, I'm just give me the aluminum can or the IV infusion. So
I did the same process with Coke. I mean, cocaine, that is. No.
yeah, yeah,
Yeah, I was like, I'd have a can of Coke, and I'm like, oh, my teeth are a bit fuzzy, and I'm just like, how much sugar is in this thing? And I'm
yeah,
like, oh, that's a big number. That's a something in the 20s or 30s. I can't remember.
yeah. No, actually, I was a lot younger, but I noticed it actually. I was hungrier during the day than if I because that constant infusion of sugar, gave me a lot of calories. But I went to diet, I'm like, man, I'm hungry all the time. But I've tried to adapt. So
Yeah. I've got just got some coffee here.
let's see here. I'm checking my um, just kind of checking the setup here. I think everything
It looks good, I
looks
think.
okay. Your face looks a little fuzzy, although maybe it's just. I mean, I'm sure. Well,
Yeah,
now,
so what it does is it records on your end and then it
okay,
uploads. So what you and I see and hear sometimes sounds less than perfect, but it looks really
okay,
good in the
okay,
finished
okay.
product. Hmm.
Yeah, I was thinking that it probably will be fine, but it probably saves a little bit
It does.
on memory that or whatever you want to whatever the computing
Yeah.
term is there. So the
if
signal will look
if
better.
it like if one of our internet cuts out, it's great because you can't tell because it's just recording locally, so you don't even hear the break unless you and
Oh,
I
okay,
start going
okay,
like what did you say?
yeah. So, well,
So
I was
yeah.
before we well, I'm not sure when we'll get started officially, but I did email a couple of the board members last night about the injectable. So I'm not sure if I have a great answer, but they said it's just kind of out of the scope. But I did email them because they this week we had the ISAP meeting, so there was like
Oh
3 days in a row of a few hours. So
yeah,
um, so I read
that's
the
great.
paper again to get more details, but then I was listening to them yesterday. They had a session on postbiotics, kind of as a it was a half hour intro, kind of what it is and everything. So it's an interesting topic, I think. So
It is.
we'll see.
I went through so many mixed feelings when reading this. Well, first I definitely had like my own misconceptions cleared up because I thought postbiotics were mostly referring to metabolites of like
Mm-hmm.
I know not all microorganisms are bacteria, but that's pretty much what most people talk about.
Yeah.
But it cleared up for me that if you're feeding like a purified short chain acetate or butyrate, that's not a postbiotic. That just it is what it is.
Yeah. Yeah.
And I was like waffling between these two thoughts of like, well, postbiotic is kind of like poetic in a way. It's like you're feeding these things that used to be alive, they're dead, and they're becoming nutrition for the new things. And then there was a more skeptical part of me that was like, is this just like when someone tries to make a probiotic and that fails?
Yeah.
But they're like, I still want to be in a biotic category.
Yeah. Well, that's what it's funny too, because there was there's other names that have been used, but or other terms that people are like, oh, we should have more terms. And Colin Hill just says you don't need a term for everything. Like you can just have a ham and cheese sandwich. You don't have to say it has to be something. So when
Yeah.
he said that one time on a board of directors meet, like a call we had, I thought, yeah, I guess that's okay. You can call it just what it is. That. Because it was after like symbiotics, you're like, oh, well then what else? What if you have this and this and this? And so he's like, just enough definitions. But
Waiting for the Sin postbiotic paper to
yeah,
come
yeah,
out.
I think that's what he was kind of saying. Like, enough's enough. Just and that's kind of what we say in the symbiotic paper too. Actually, is if you have a probiotic and a prebiotic, and if you can still say you have a probiotic and a prebiotic, you don't have to say symbiotic if you don't want to, and if you. And unless you've tested it, really you shouldn't say it's a symbiotic unless you've tested it and shown that the combination is effective, But like like they said too, on most of the time you want to check their safety, but you would hope there wouldn't be any antagonism or, you're not going to put combinations together where you think, oh yeah, this nasty compound's going to be
Right.
produced based on this substrate, or so it shouldn't be a huge huge risk area, I don't think, but it should be tested, always. But yeah, so we'll
Yeah.
see on the regulatory side. I'm not sure how much I know, with that, but there are some good things to talk about there, I think too, though. And actually, they were saying, at least in Europe, it probably applies to the U.S. as well. It's probably easier to get approval for something like when it's inactivated, because then they can't, the thought of the safety risk of taking over and causing some infection or something. If it's inactivated, then it's less risky there. So that's the thought anyway. We'll see what the regulators do, and how that goes. But anyway, it's an interesting area for sure. And there's a lot of activity. So I'm not sure if you guys are working much in this, like with Traverse or not, but I don't know. In the pet side, it's certainly of interest, I think.
Well, yeah. Well, I mean, and you and I had a call with a unnamed client on this, and I think what they have is actually a postbiotic if it confers a health benefit and meets all those definitions.
Yeah.
And we were trying to get similar products from competitors or other companies, and I had just kind of a little email conversation with one of the people because they were selling something similar, and they had recently called it a postbiotic or made a switch. It used to have a different name, and now it's a postbiotic. And it's kind of curious. It doesn't seem to me like a name that it seems like it has sort of arisen just out of the community. Like, did some just like marketing person suddenly come up with it? I don't think that's the case. Because I don't know. If I had to come up with like what would be like a great name to like wrap all this up under one, it wouldn't have been the word postbiotic.
Yeah.
And he's like, well, we've been in this space for decades, but now they are, probably to their benefit, will kind of reframe it as this postbiotic product.
Yeah, I think that's true. That, and it is good because I know there have been several. Some people were saying like just a dead probiotic or a ghost biotic or other terms were used, but if you have something that at least if most people, even though it's been misused by some people, is the collective term, then people kind of, I guess, realize what you're talking about. So hopefully it will. Hopefully the term will catch on. But yeah, others will argue when you have even Glenn Gibson said even prebiotic, he wanted to call it something else, but his kind of collaborator convinced him we should say prebiotic because he said prebiotic is like you think of prehistoric like dinosaurs or but there was another argument to that of why a prebiotic is a prebiotic, and even today he at a recent conference he kind of said that. But but again, the postbiotic, I think it's because it's after maybe they've done their activity, I guess is maybe why it's post. But I don't know. Hopefully, just people grab onto it and then go forward, and we know what we're talking about. But
based on those like PubMed hits in the ISAP paper, it looks like it is the new term. So it is officially caught on. So. Someone comes out with a different paper. It's like the same thing. We're gonna put our definition out. It's like, dang it, we got scooped. Yeah. Well, post scooped.
Yeah, no, that's the thing with some of these, even prebiotic and symbiotic. You go through these. I've been on a couple of those expert panels, and you arrive to a consensus, but there is more discussion than I guess I would initially thought there would be on. I guess in part of it you have to think about what your intent is, and then if there's any way that like an antibiotic or some other substance can come in underneath that definition. So it is you really have to be careful of that. It's just so something random doesn't fit for the term, I guess, and kind of that wasn't the intent. So
I guess it is difficult. How So I'm sure we've both been on various committees for these things. To me, this whole task sounds like it must have been like herding cats, right? Get like more than two scientists to agree on a term or something. It's just like, I don't know if I want to step into that. But at least ISAP, you guys seem to continuously be coming to these consensus opinions on these. What was the work in progress to get to this point on postbiotic? What was that like?
Yeah, well, I didn't serve on that committee, but I can tell you what the committees are like. And you're right. Oftentimes you have about 10 experts, and I always say you have 10 experts with about 11 different opinions because someone's going to be flip-flopping here and there, and that sometimes is me. But it's a long process, and to you really, all of these, luckily, all of the from probiotic all the way to now fermented foods and postbiotics that were more recently kind of just came out in publications earlier this year. But it's a year and a half long process, but an in-person meeting. Luckily, it was all of those were at least met on pre-COVID. So, but it really takes a lot of discussion to do that. But there's a lot of prep work up front too. So when I served on the prebiotic consensus panel. I chaired the symbiotic consensus panel so I learned the process at a different level with that, all the way from A to Z, like all the way from starting out to then getting that last author proof. It was on me and a couple other key people, on the committee. But really, you kind of have to give people marching orders and come prepared to discuss a certain thing or present a certain thing, and then we really discuss it. So it was, I think, all the panels really, it's about a day-long, full day meeting. Usually the beginning is kind of presentations of different aspects of it. It can be talking about the mechanisms, or when it's pro or prebiotic, what's the substance, how do you measure it, how can you document this or that, and then really so the definition is important, but then the use and all the aspects of the implications of it, when it's regulatory or just production, considerations, I guess, are all discussion points. And then really people get their marching orders again, say, okay, here's the main structure of the paper, and we're going to fill in the blanks. And then the symbiotic was much more complicated, because we had kind of the microorganisms and the substrates together, and so I think it went through more versions that is typical. But even with the postbiotic consensus here, I think that even listening and talking with the committee members, you ask a certain question, you'll get slight different answers, and you have to be reminded, well, remember this, remember that. And so that's why I think these consensus papers really try to touch on all the potential questions people would have, and sometimes they're written as that. The probiotic consensus paper, it was kind of written as, this is a question, here's our response, here's another question, here's another response. And so they're kind of written in that way, and that really is there to help all the stakeholders, whether it's producers, regulators, the media, other scientists. So we're hopefully using the same terms, using them correctly, and then just again, all the potential questions that come up, we can hopefully be addressed. And you can't think of everything, and it'd be way too long, to address every single question. But hopefully the main ones are addressed. But yeah, it's quite a process from beginning to end.
So I guess you've then been a chair and you've been a member. Would you chair one again?
Yeah,
would you either contribute or lead?
Yeah, I would, but it's certainly I being on a previous committee, before, I kind of knew what was involved. It was a lot of there was a lot of discussion, even all the way through. By far of all my publications, this is and I was the one in the final end. I was looking at every author proof and everything, so I was doing much of that. Again, there were a couple other, like Mary Ellen Sanders and Glenn Gibson were really the two key people that were there most of the way. But so it went through a lot of revision, and so that was exhausting at times. But it was I do think the final product was worth it, and so hopefully it addresses a lot of the questions that are out there. And so I would do it again, but it would have to be the right timing, though, to make sure I have enough time to commit to it, because it's a lot of work. So
absolutely, yeah. They're great papers to have out, and I'm sure they get cited a ton. So many. It becomes the new introduction section for a lot. It's like, well, the ISAP says this is what it is, and this is what we're doing.
Yeah.
The symbiotic one. You have to come to an agreement, I guess, on both of the previous terms, which you guys had already done. But I can see how that one gets maybe exponentially more difficult. And you mentioned measuring them, and measuring prebiotics is difficult. I mean, I didn't really understand the complexity of those. Whatever, it's like a carbohydrate chain. That's cool. We can measure those, right? And even for individual types, like we did a project on pectin, which I thought was a fairly boring inert substance, which is wrong. And I thought, well, pectin is pectin. And, oh my gosh, there are so many different chain lengths, branching, residues on air. So to then combine that with the seemingly infinite amount of different types of prebiotics you could have, couple that with different probiotics to put that into a symbiotic. And what's interesting that you just sparked in my mind is maybe for some people that ultimate product acts as just a prebiotic for them or just a probiotic for them. And maybe for others, it really is the combinatorial thing that is beneficial for them. So now you've really covered your bases there. And what's ironic about postbiotics is I remember people saying like, oh, you're taking probiotics, like that stuff is just going to die in your stomach, right? From the acid. And now it's like, well, that's okay because maybe their components will still have a postbiotic effect. Like it doesn't matter if they're living or not. And the thought was like, well, you only need to seed a couple of bacteria. So it's like if you can take those billion, whatever that 10 to the whatever exponent 9 CFUs, as long as a couple get in there, you might be able to seed it, but that's not really the case. Well, okay, if it is a probiotic, yeah, that's probably the case. But you can now fall back a little bit, and perhaps it's still beneficial.
Yeah, that's where there was discussion even before the postbiotic term, and really kind of when people started thinking about, okay, there could be another product category here. I think there were people showing evidence that even if there were at the time, I think they're calling it dead probiotic, where it wasn't the viability wasn't there, but there were still some of these like you get into some of the terms microbe associated molecular patterns and touching on receptors in the gut, and whether it's immune related or something metabolic related in the epithelial cells or some immune cell. There was some evidence there, and people are like, well, but they're not viable. So how are they functioning? And so now, I should clarify, the postbiotic is supposed to be more than just a dead probiotic. There's more complexity there. But you're right, though, that it kind of touches on some of the different mechanisms. It's not just a live bacteria that it doesn't have to be just a live bacteria that has a specific function. It can be other things, and so it is kind of. And I think because of these advances of the past, couple of decades, really, I think that's where ISAP initially, and this was before I was on the board, thought we need to kind of update not only the definition, but it's just as much of the definition. The definitions have been adjusted a little bit, but the intent is basically the same. It's how do you measure it? Now we know so much more on the molecular side, and we can measure things we couldn't measure before. Measuring efficacy and stuff, I think really was for me anyway, I think those are the key parts of those papers where you can say, well, there's new methods now, to measure different things, and potentially on the production side as well. You can just get more specific, and you can do, now all of whether it's probiotic or the microbe of a symbiotic, or even for the postbiotic, characterizing that microbial component, you can just do whole genome sequencing and get all the information, that you couldn't do 20 25 years ago. So it's pretty amazing what you can do, and that's kind of the expectation now. You should really know what you're working with, and even the postbiotic, the end product, you're probably not going to be able to do that, but you have to know the strain or strains at what you start with, and you have to characterize that, or that's the expectation. And then also the strain is one thing, but how you the fermentation process, or how you're producing that, what you're feeding it, the time and the temperature and all the other the things when it comes to production is pretty important to describe that. And that's really key. Certainly I've talked with companies before on that the secret of the strain that they have, that this is the genome sequence, this is the specific strain, and that's they don't want to share that information.
Right.
But just as important, maybe I would say, just as important probably, is those growing conditions and what you're, the process there. I think the protocols that are in place that have to be, important, because you can start with a strain, but depending on what you feed it or what, the conditions, you can completely change the profile of what that postbiotic will be then. Just like a probiotic as well, what you've been feeding it, and, that at least initially when a probiotic hits the gut, how is it set up? What has it been getting, and how, there'll be probably a lag phase there. But the postbiotic as well, what profile do you have on the bioactive, metabolite side, or even probably the receptors of those, bacteria now, or yeast that you've broken down or at least inactivated some way, but that's probably going to affect their activity as well. So, and even I don't work in the production side, but even the inactivation method, there's someone at a recent conference is talking about autoclaving versus pasteurization. Autoclaving, some of these things you could certainly inactivate that way, but you might then destroy a lot of these bioactives that are there, where you might want to pasteurize instead. Or there's people have showed, in the paper, even the consensus paper says, modifications of spray drying or different.
Right.
There's a lot of different methods you can use to inactivate, and they may or may not affect, the activity of what you have, as that mixture then. So you have to be careful. And so really, I think a nice thing about postbiotics, there's a lot of opportunities there here, I think, from an innovation perspective of, again, starting with the strain, the production procedures, the inactivation, and there's a lot of steps there. And for some people, it's in the consensus paper too, they highlighted on intellectual property protection. There's right. ways you can protect, some of that as well if you're a producer. So
yeah, I thought that part was interesting too. And you can't always have it both ways, because you want to have intellectual property protection. I think a lot of people don't realize that when you do submit a patent, you have to describe in great detail what the thing is, such that someone else who is, quote, skilled in the art can figure out how to make it. So you're not there isn't a secret when you publish a patent. Like, it's out there now. Now it's defensible, and if someone encroaches on that, you have a legal right to pursue litigation. And it's kind of like, well, if you don't want your secret sauce out there, you probably shouldn't patent it. Of course, if you're a young company, you probably are going to need to have a patent to get investment, or show that it's protectable. But the reverse side is like, well, maybe it could be your trade secret. But then if it's your trade secret, you sure can't really put much on a label. People are like, well, how does it do what it does? I don't know. Can't tell you. It's a trade secret. And in the paper, they have that section on, you'd want to at least provide aspects of your product to a public repository, whether that's like the genome, or something else. But as you talk about all these production factors that go in play, it's really kind of mind-boggling. I mean, when I think about when we would design experiments in grad school, you do your power analyses, and for what we were doing, 12 was like this nice magic number. It was a good trade-off between like cost and logistics versus adequate stats. But I'm just thinking of like, when you're doing your ANOVAs and you have all your effects, like A times B times C, and they all have different levels. It's like, wow, when we do these reviews on products these days, it's like, well, we think this is important, but we don't know much about how long you have to consume it for, the dose, and those are like the two main things. Now it's like, okay, well, how is it heat treated? How, you have so many different ways of production. And this is where we get tongue-tied ourselves working with clients, is you have like multiple people all kind of like pointing at each other. It's like that Spider-Man meme of them all pointing at each other. And people who are in production are wanting to know, maybe from nutritionists, like, well, how should we produce it? Is there something that we should try to preserve about this or enhance? Nutritionists, I don't know. People haven't really made this thing before. So we have to test it a bunch of different ways. And then you might be selling it in multiple different regions or countries. And so you have to know about the regulatory considerations too. So it's kind of mind-boggling and sounds like an obstacle, but a better way to put it is, as you said, there's a lot of opportunity. And especially there, I guess, on the IP side, is if you and another company or maybe multiple companies could all be working from the same strain, maybe even, but your different preparation methods allow you some uniqueness to what it is. And that gives you, so much more flexibility to innovate around it. I just think about how much work would go into it, and they're like, all right, we know how stable it is. We've characterized the components of it. Let's finally run our clinical trial. Null results. Nothing happens. It's like, which of those do you do first? Do you get kind of like a dirty version of an unpurified thing, see if there's benefits to it, and then kind of backtrack, purify the right components, or do you try to do a lot of the production aspects first?
That's a great question. And I'm not sure if I can answer all of that, but I think there's a lot of different. I'm not sure if there's just one way of doing it. I think some of the existing companies, I think have, they've, this might be a byproduct stream where it's
Yeah.
at least that's the way it started. Where, oh well, we've been fermenting to produce X, and now we have this product here. What do we do with it? But I think I think there are some of the existing that maybe started that way, but I think now there are people that, or I know there are people that are screening. They'll use, in the end, you need to have that target host, just like all the biotic definitions. You need the target host. So if it's a human, if it's a dog, if it's a cow, whatever it might be, you need the target host, and really the life stage or the health status of that host as well. There's a lot of in that kind of, you can get into the regulatory side there, but what's your target? But to start out with, I think there's a lot of screening done in vitro, and now we have, there are ways you can screen it in vitro with maybe fermentations, maybe with enteroids, things like that. If you're looking at how is it being fermented, or how does it affect the microbes, how does it affect maybe some, or an in vitro cell culture, that. But also you can, the nice thing with the omics technologies, you could do a metabolomic profile of this as well to see what do we have here. And I think there's a lot of screening tools that people are using. But I think it goes both ways. I think a lot of them they might already have the other thing too. You can innovate from, we have this process. So this is our product A. This is what we have already. How do we make product B? And that's where, there already has they already have the production side set up, or then let's modify this and this and this, like in a pilot facility or something, and see how we can modify it. Like you said, maybe using the same strain, or, if we just know here's a novel strain, and we'll it's a little bit different process there. But you're right. You could start with the same strain and make completely different products, probably with, the gene, the genes that those bacteria or yeast or whatever microorganism you're working with, whatever they possess. And so it's, like you said, you have to disclose some of that information if you if you start patenting it. And even like you said, you can't just say, oh, it's magical, it does this, and we can't tell you how or why. You have to get some, people are going to want to know at least some of the mechanisms or some of the bioactives, or some reason. So you have to disclose some of that information. And like you said, that some repository for the genome sequence typically that's kind of the minimal thing you need to do, to do that. And so again, it shouldn't really be a secret. I think the secret is really in the processing of it, the fermentation conditions. And so there's probably some of that when it comes to the probiotics as well. But with postbiotics, it is it's at another level of complexity. I think, like, there's in a probiotic, you can remove a lot of the other things, and so you have that viable bacteria. But here, you probably want all those things it's been producing, and modify, how it's been modifying the strain or the strains that are there. So, but yeah, that's the kind of the playbook there of how to go about it. There's a lot of, I think, things to consider, and probably many different ways of doing it. The nice thing though is that I think we do have a lot of different screening methods now that can be used to innovate and again, provide a little bit of positive that allows you to get into that in vivo. Even you might even start, again, with a lot of people start with rodent model or a pig model, or for a human or something like that, where before you go into that clinical study with people, you want to know safety certainly, but then even you have a chance of efficacy, dosage. A lot of those things have to be worked out before you go right into the clinical study. So, and the same thing applies to pet. We do a lot of maybe not animal models, but a lot of in vitro testings oftentimes before we go into the in vivo study, just so you're sure that you're in the right range from a dosage perspective, and because that's important from an efficacy perspective, but also the cost. That you can pass on some of that. If it's a really effective product for whatever your target would be, that's great. But if it's just the dosage is way too high, it's just not going to work. So you have to know kind of where you are and how it fits into the structure of or the cost of the product. So and whether that's a food or a supplement, or there's ways
Yeah.
of that can change things as well, or affect how it's sold, and how it's really the whole process, I guess, if you think about it. So
When when you're doing in vitro work, are you typically taking like in vitro fermentations? You're taking fecal samples from your host of interest and spiking in the product and looking at fermentation rates, probably microbial diversity, things like that.
yeah, that's typically when we do in vitro, that's what we've had set up in our lab for a while. And they're kind of closed cultures. We get fecal inoculum for like you said, the target host. So I always say anything that poops, we can do the in vitro fermentation with it, because you can collect that. And usually we have a few different subjects or donors, I guess, and then we can do those fermentations. But yeah, we prep the media ahead of time, and whatever substance. And again, most of the time I'm working with prebiotics and fibers, but you could do a similar. Really could be almost anything to see how does that affect the microbial population, what they're producing, things like that. Again, the other, I guess, category kind of in vitro are kind of the cell types and enteroids and things like that, which we don't really have up in our laboratory, but there are labs that do. And that's kind of looking at, if you had this substance or this, this microorganism, what would it do if it, directly was contacting a colonocyte, or something like that. And so you can, and that, I'm not an expert in that area, but it sounds like there's a lot of innovation in that area too. These enteroids that can mimic different cell types, and they can.
Mm-hmm.
A lot of the signals they're sending are similar to what would be in the gut, and so you have to. All these in vitro assays, you have to take it for what it's worth. It's only probably predicting what would happen in vivo, but still, I think you can learn a lot using some of these techniques. So it can be quite useful in making decisions before you get too expensive and too complicated.
Yeah, there's so with pre and probiotics, for example, and I only give these out because these are the ones I know on like the brain side, which is there's a fair amount, I guess fair relative to itself, more than I thought there would be, of evidence that even in humans taking pre and probiotics, they can do things like relieve anxiety, relieve depression. They almost have these drug-like functions. But if you were to, and we know that those same things can change, who is in the gut and the different populations there. But I would never think, the link between those two is really vast. It's like a vast chasm. So it's like if you were to take this bottom-up approach of who is this product feeding, what are they doing, what are their metabolites, their genomes, I'm thinking you would never get to the point of saying, let's use this for a treatment for anxiety or depression. And maybe that's the magic of having so many different institutions researching the same thing, is you have people taking a top-down, bottom-up approach. And I'm thinking here mostly from like an industry perspective of, if you're a company, how much mechanistic work makes sense to invest in, because you could just be doing that forever, and spending millions and millions, and then one day maybe a light bulb goes off and you're like, all right, we found the last link in this pathway, it's going to be used for this thing, and meanwhile we're like negative $1 billion in the hole in the that would never happen. And on the pet side, how do these things typically come about? And you mentioned companies might have existing waste streams that they're trying to upcycle, create value out of. What is the split between, in pet food, companies saying, well, we want to make something that does X, it improves the health of the animal, their skin, or helps alleviate the aging process, versus taking this like ground up approach, looking at mechanisms and kind of like pinpointing a purpose for an ingredient?
Yeah, I think that's probably a lot more top down to see. And a lot of our research is kind of that way. We've done some safety testing, but typically you want to see, are you able to measure an outcome that shows a benefit? And that's, if it doesn't, then you can,
Full stop.
hopefully you're looking at different doses or something like that, and measuring what you think. Sometimes you're maybe measuring the wrong thing or at the wrong time. So there's some discussions there, but oftentimes you want to make sure you have an effect first. And then if you can understand the mechanism, that's great. I would say, especially like, if they're a startup or a smaller company, typically they're not going to invest in the how. It's just if it has an effect. But there are the larger companies, I think, that are, been around for decades and they have large R&D programs. If you can ever get to identify the mechanism, then you have knowledge and I think in your corner that others maybe don't have. And especially if you don't have to disclose it right away, you can develop other products, and that can also feed other things, I think. But up front, initially, especially with a novel product, I think typically, like you said, it's kind of a top-down approach of, well, we have this, or there's a lot of sharing as well coming from the human side. Dogs and cats used to be used as animal models, and they still are in certain, probably in pharma, but in nutrition, now with non-invasiveness and stuff, you're not really using dogs and cats as a model for people. But there's a lot of sharing of information, and I would argue that the human is just as much of a model for a dog and a cat now, because they'll see, or even rodent models, that something was shown, can we apply this to dog, or, and oftentimes these not that there isn't a lot of innovation just on the pet side, but a lot of it comes on the heels kind of some new concept or new product on the human side, and it quickly moves over to, let's try this in the dog. Does this have application? Like you said, it has to be the right target. But there's a lot of potential. If you look at all the structure function claims in the pet food, whether it's the foods themselves or just supplements, targeting joints or skin or digestive health, immune system, all those structure function claims are, well, many of the mechanisms, many of the ingredients, and the claims are very similar to that of humans. And so I think there's a lot of sharing information between humans and pets at this point. And a lot of the big companies are, collaborating with or are owned by a large company that's in the human food or human care space. So, but yeah, it's interesting in academia. There's both approaches, and in a lot of the bigger companies, there's both approaches: the top down, bottom up. And if it's published or if it's shared, you can, the whole community can benefit. But there's a lot of that's kind of held, and I understand you need to keep your competitive advantage if you can, and so some of that information is not always shared. But and that happens, on the human and pet side, livestock as well. I'm sure there's some of those things they want to keep secretive, and so they have an advantage there, or hopefully they have an advantage with new product development. So
yeah. Well, and I've heard too of companies developing things and humans being their primary target, and they're like, well, if we can't get approved for this, we might go into animals. Maybe it's an application for livestock or for pets, kind of like this fallback. I don't know how that makes people in pet food feel. Oh, are we just a fallback industry? Or if that's just a legit good strategy to take. And I mean, on publishing there too, I think if you are developing something new and innovative, you definitely want to work with your lawyers and get protection, file a provisional patent application. But I wonder if even publishing the neutral or negative results could be, almost defensive, in the sense of, you're like, hey, well, we thought we had this special process or type of postbiotic, but it doesn't actually do what we thought it did. Publish it, and that prevents others from trying to make similar claims when you've done your research and you show that it doesn't work. Of course, multiple groups can of course publish on the same thing and find different effects. But from a patenting standpoint, it would at least reduce the novelty of those things. It almost makes sense. So, okay, we've got all these omic tools that we could use to try and look at efficacy. Well, let's come back to the definition, actually. Just center this conversation for a second. We've gone this far without actually talking about it. Okay, I pulled it up. Not going to say it from memory. I probably could almost. So it's a postbiotic is a preparation of inanimate microorganisms and or their components that confers a health benefit on the host. There's a lot in that one little thing to unpack. I feel like the most complicated part of that is confers a health benefit. But, someone who makes these might say, nah, production's more complicated. And in that paper, they talk about all the different mechanisms that might be at play with these. I struggle so much because it's like, well, none of those mechanisms will tell you if it confers a health benefit. Well, not that's not entirely true, but it's not always clear how fermentative metabolites lead to some health benefit, or how upregulation of immune-related genes. Okay, these cytokines changed. Did that actually reduce susceptibility to an infectious disease or change the immune response? And so that's just a harder thing for me to wrap my head around. I'm rambling. I don't even know what my question is here.
Well, I know one thing we were talking about before is what do you measure? Because you're right that you have all these potential mechanisms that it might be a means to an end, where this is okay. We think it goes this way, but oftentimes you can't measure it in a person or in a pet. Usually, if you're not being invasive, so you kind of need biomarkers, and that's the good thing with I say in vitro, or even in other types in silico type of modeling and proposing how this is working, and all the research that's going on in this area just in general will identify these potential mechanisms. And if you can have biomarkers, then you can potentially measure. But it is hard. And that's all of the all the way from probiotic to postbiotic, that health benefit in the target host. It doesn't have to say the target host, but a health benefit is a key thing. And it, like you're right, it's very difficult to measure that sometimes. Of how do you know it's a benefit? The other thing that gets involved, I guess, that I would add on to that is, if you have if you're trying to support a structure function claim, you can't get into a disease or anything like that. So that's the other thing of most of our research in our lab and a lot of other labs is you're working with a quote unquote healthy population. Well, how do you show that a healthy population is now healthier than they were before? So that's always the tricky thing. And as far as, and they even touch on in the consensus paper, if you want to go down the medical food or medical device or some other avenue, you can do that. But a lot of people don't go there. They're looking at a food additive, or they need GRAS status or a dietary supplement or something, where you have to be really careful with the claims you make. But how do you, what are you going to measure? What outcomes do you measure? And then do people agree that that's yes, that is a health benefit then, or no? It kind of comes back to FDA and the fiber definition, with short chain fatty acids and microbe changes aren't themselves a health benefit, which I would argue, I think they are, at some level, but and other people will argue that as well, I think, but they're kind of biomarkers of what may be beneficial. So it's a tricky area, and here it's actually more complicated because it's with fiber, I guess it's more complicated as well. In prebiotics, it's not just short chain fatty acids, and that's what has been the most studied. But there are other bioactives and other proteins and other things that are made. But that's kind of a work in progress, I think. So people kind of lean back on or use the short chain fatty acids, but they can be used as secondary messengers, and other gut peptides are stimulated by them, and barrier function is can be improved with them. But those changes can be affected by other things as well. And so that's where I think what's really exciting in the postbiotic area is that we're kind of, it makes it more complicated, I guess, but there's, you can touch on a few of these, probably more of these mechanisms than just a probiotic or a prebiotic by themselves, I think, because you have a complex mixture of it. So, but yeah, circling back around to how do you show a health benefit? That's what I kind of pound my head against the wall on many days of how do we measure this in a dog? Can we measure immune function or gut health? What markers can we measure? And it's a complicated area or a complicated thing to do. And many of the same complications are there with humans, although at least a human can tell you how they feel. An animal can't really say,
do a survey.
I'm feeling more bloated today. They don't say that. So it's and even if they're not feeling well, they often will hide it because that kind of shows
Mm.
a sign of weakness. So I think it's just kind of ingrained in them not to show a problem, and so it's a little bit more difficult, I think, on the pet side. And livestock, I would argue sometimes it's easier to show there too, because you can, there, if you're showing, looking at rate of gain or efficiency, or it's a different way of
Yes.
raising the
Yeah.
animals, than a pet. So it's more production and morbidity, mortality versus kind of health and longevity on the pet and human side. So they're even a little bit, the
Oh
pets
yeah.
are kind of in the middle there somewhere.
That is interesting. Yeah, you can very clearly on the livestock side. I mean, if what you make doesn't provide an economic advantage, who cares? Um, and companies, they'll still invest in mechanistic research, as you've said,
Mm-hmm.
and make spin-offs of things they developed. And on the human side, I think if you can at least convince someone that it works, yeah, half of it might be a placebo, but I guess the thing still works.
Mm-hmm.
Not for everything, of course. And interesting, yeah, much more difficult in pets not being able to speak.
Yeah.
And also money's not the driver there. It is just the health and well-being from probably a pet owner's perspective, or a veterinarian's, of course. There's
Yeah.
objective markers there too.
Mm-hmm.
And okay, what I was gonna say just went out the window. That's just it's gone, that thought. Oh, okay, there it is. Yes, so you're right. There is more than just short chain fatty acids. I mean, there's only like 6 metabolites a bacteria can produce. Now there's so many. And when you and I had met before this on that other project, it was, how can we measure gut barrier integrity and those things. And what's interesting is maybe the easiest, the most well characterized biomarkers might be the most boring, in the sense that it's the things that have been easily measured for decades that people have been doing, but those might not be the most effective methods to use. So if you come up with a new method or something that's more sensitive, it's like, that's great, but it's almost like tough luck, because you can't fall back on these decades of research of other people using this, and it's well known and people understand how it works. But I do think I think a lot of data is kind of left on the table in some sense. And you know what? I was always jealous of people who did clinical research for this reason. Like in humans specifically, these big cohort studies, I would get so jealous of grad students who got placed in those labs. I'm like, all you have to do is like mine someone else's data set for your 5 years. You don't have to see a subject. You don't have to design anything. You just have to slice and dice this thing 1,000 times. But I think that approach, though, of like data mining could also be done, and I think that could be really effective for the biomarker approach. And I tried doing that at the end of my PhD with short chains specifically, and we had fed a lot of different types of prebiotics, mostly oligosaccharides, to pigs, and I had also collected some brain outcomes on some behavioral cognition outcomes on a lot of them. And what had surprised me is, well, maybe not always surprising, but short chains didn't always go up. Sometimes there wasn't a noticeable change, but the relationship between those short chains was very sensitive to the diet. So sometimes it was related to a behavioral outcome. I could show that, for example, a reduced butyrate was associated with better memory in these pigs, but that wasn't the case on diets 1 and 2, but it was for 3. And that's where again, maybe that just adds to the complexity, but I think there it does give you some opportunity, though, to show in what system or environment do those things become biomarkers of what you're actually looking for. And that might help create a stronger case for your product eventually. You might say, well, individually, all of our studies maybe had a smaller sample size, but together you take that meta-analysis approach, and not just meta-analysis in the sense of, you qualitatively say, well, look at, if we qualitatively look, these things went up and down in X number of papers. Like, no, literally put all of that into like one Excel sheet, do the correlations and regressions, all that stuff, and then figure out if you actually have a biomarker for that or not. I think that's becoming a more common approach as people become more and more data literate, and these things get easier to do, which then means if you're in it for the long haul on your ingredient, maybe you do want to have some common outcomes on a lot of your studies.
Mm-hmm.
Gut barrier related stuff might not have worked in one study, but as you've mentioned, maybe it was a dose, or maybe it was form, or something like that. That's not a very good argument, though, to make. You're like, well, it didn't work last time. Just kind of do it again.
Yeah.
Just keep going until you find something.
Yeah, I do question. Sometimes I'm like, well, how, with these fermentation procedures, can you have consistency? So you are delivering the same components every time and things like that. But on the, I don't want to say method development, but maybe biomarker discovery side of it, I do think as a community, with now people have gone to metabolomics and untargeted strategies where in our lab we are always targeting the short chain fatty acids. We'll look at some of the protein catabolites and stuff, but that's what we've measured for the past few decades, and even before I was in the lab, that we were measuring. The others were measuring some of those metabolites, but once you get into on the metabolomics side too, I've served on some advisory panels in the past and just discussed, well, if you're talking about fecal samples, that's one set of metabolites. But if you get into the blood or the urine, well, then it's so complicated. But you take what is initially produced by the microbe, but then it might be modified. It can be, added, methylated or sulfated, or, affected somehow, or something's been modified. Now it's in the bloodstream or it's in the urine. And, what is it doing, or what did it do if it's in the urine? Where's this coming from? And so there's a lot of those that you can identify. We've had a couple data sets like that where, just feeding like a high carbohydrate versus a high protein diet, we saw this huge, divide when you look at a, principal coordinate analysis plot of the microbiota, or a PCA plot even of the metabolites, and these high protein, and this was cats, but they completely separated. The same animals just fed a different diet at a different time, and they completely separated. And a lot of what the distinguishing factors, and especially in those high protein fed animals, were basically co-metabolites of the microbe and the host. So this is blood metabolomics. So it's pretty interesting, actually, of whether it was better or worse. We didn't really test that, but it easily showed you can really affect the serum metabolome. And others have shown, whether it's fecal or urine, the same thing. And these are all in healthy animals, and you can show this great divide. And it kind of speaks to, if you're looking at heart disease and cholesterol or something, we've, on the human side for years, and somewhat on the pet side and livestock as well, but we have biomarkers of certain things. But I think the technology now allows us to go at a much, we can actively search for it, if we want to. And hopefully, in the end, if you can have a handful of biomarkers that tell you something about, gut barrier function or immunity, or, something like that, that's really, decades from now, hopefully researchers or, producers, manufacturers have those biomarkers that say, oh yeah, 50 years ago, they didn't know they didn't know this thing was right in front of them, and they didn't know how important it was. And hopefully, there's some of that discovery, and we can start, adopting some of those things, and then be targeted. At some point, if you can be targeted, that's the key. I think that it's not so expensive, and not so, it's so data heavy with these big omic data sets that you're trying to mine it and trying to find what's really important, but it's really difficult to do. And even if you're properly powered, and the people on the bioinformatics side, they're always wanting, we have all of these metabolites, but then an N of 12 or 20, and they're like, this is completely wrong. You need thousands of, samples or something. You're like, well, that's not what we have. So it's, it's difficult to do, but there are people working on that now with, and it is interesting too, if you have industry or government, funding sources are kind of all looking at it a different way. And I think that's building the collective database, to a greater extent, that I think it eventually will help. But a lot of these things, like you said, the gut barrier, if you had a new biomarker, one, you'd have to convince others that this is effective, and then having people adopt that. And hopefully it's something that's not, too complicated. Once you know what it is, it's an easy way to, measure it, and so people will use it. But especially in the gut, there's a lot of, it's like a big black box, that it's, how do you know what's going on? And how do you know it's stimulating, gut immunity or, whatever it might be? It's really difficult to do that if you're limited to kind of urine or blood or fecal samples or saliva. There's some other things you can do, but it's really difficult. But that's where I hope there's certainly at some level you need to get to your target host, but I'm still hoping that a lot of these biomarkers and things from rodent studies or other animals can be, used in other species as well. At least so hopefully that will be there. One key thing that is lacking that people have said too is, the metabolomics databases are very host-centric, very human-centric. So you think about the KEGG pathways and metabolism in particular, it's all about the human, metabolism. But really in some of these, we really need a database and a data set that's focused on the microbes. What are the microbes doing, and are they keeping it internal? Are they secreting this into the lumen where other, the host and other microbes can cross feed, that's kind of an unknown area. But I think there are companies working that field or in that area as well to build up the metabolomic database for the microbes, not just thinking about humans all the time, that, and some of it will be, there's common metabolites. They'll probably have similar, feed similar pathways, but there can be completely different pathways. We know the microbe, genomes have, 150 to 1, numbers of genes versus what our genome is. So they can do all kinds of things we can't. And so it is, and then of course produce metabolites that we can't produce. So I think there's, that's a whole area that over the next, decade or two, I think will be really interesting to kind of follow and see what comes of it. Because that'll apply, a lot of that mechanistic and kind of base level molecular work probably doesn't have to be done by the industry, but I think if people are watching that and pulling out, interesting points of data, then they can start, they can apply that to say, oh, here's how we're going to use that. This data set's out there. Let's measure this, and this, and then we can go forward and from a health perspective, we can make more claims about that. But so it's, on one hand, it's a complicated and confusing and sometimes frustrating area, but on the other hand, there's so much potential here. It's really finding that needle in a haystack is usually what it is, that key biomarker or a handful of biomarkers that really is going to be important. And sometimes, like a lot of things, it might be discovery by accident and something's found, and then wait a second, let's do a little bit more. Let's do another study on this, like a targeted, and that's how things are discovered. And then it's, it kind of breaks things wide open. So it's an exciting area, but sifting through the big data, I go home a lot of days. I'm just exhausted, like mentally exhausted. I'm not smart enough to figure all this stuff out and interpret it. It's, but at least others are working on it that are smarter than I am, so we can try to apply the information. But that's the other good thing about this area. It's a very collaborative area too, because you can't be an expert in everything. So you have the ecologists, you have the people on the biology, or the microbiology side, the nutrition, is right. In my world, nutrition's in the middle here because it affects all these things. It depends on what your viewpoint is of the world, I guess. But there is all this, it's all interacting. Nothing's on an island anymore. They're all these systems are in these tissues are communicating with each other. The microbes are communicating with each other and the host. And, it's, if we could understand the signal, and that's the language that's being spoken is what we're trying to figure out. And it's really difficult to do, but it's job security, if nothing else.
Guys, this is so complicated. We need more money to research it.
Yeah.
The collaboration aspect is important in there. I guess you don't even have to collaborate. Maybe you just get out of the zone that you're normally in. And when I first started in the lab, I was looking at all this pig behavior stuff, and I'm like, no one's ever done this. This is so unique. But I was using more psychology, neuroscience words, and those are not what's used in the world of animal welfare. They would do the same exact thing, but they just had different names for the tests and things. And it took a bit of a review until I found all those. And I was like, oh, okay, no, this isn't actually novel. Other people have been doing this. Oh, darn, my idea wasn't original. But then at the same time, it's like, okay, good. I don't have to do that experiment anymore. Yeah. Someone already did it. And on the biomarker side too, well, yeah, this is where publishing is getting archaic to me, because there's only so much room you have to talk about these, about the results that you found. And you're going in with presumably a hypothesis or at least some thought on what you thought you might see. And not everyone does this, but some people are like, these are what our primary outcomes are going to be. These are secondary. Or if it's not explicitly stated, it's just kind of like inferred by what the author talks about. And there, I'm positive, is plenty of stuff in supplemental that is someone else's primary outcome. It wasn't yours, but that's exactly what they want to see. And I get really bothered by editors sometimes when they're like, you already put this in a figure, don't put it in a table. I'm like, someone might want to know the legit number and standard deviation and stuff like that. Like, why don't we have better systems for sharing data and things like that? But back on the biomarker side, in some ways it is really simple. Like you can look at, on a safety or toxicology perspective, organ weights, for example. Most people wouldn't call this a bioinformatic approach, but just saying, well, what's the relative organ weight compared to body weight? And people do this for cholesterol, LDL, HDL. What's the ratio of this to that? Acetate, butyrate, Bacteroidetes to firmicutes, things like that. And we did this big meta-analysis on an organ weight, which is why I bring this up. And what became interesting is looking at absolute values wouldn't always pick up on an abnormality. Intestine weighing 300 grams or whatever it is. The relative wasn't interesting, but if you regressed it against body weight, animals of a specific size should have a relative intestinal weight of whatever number. And what this allowed us to clue in on is, I guess, getting back to like the context. What is a expected value for a given health status, for a given age of that subject, for a given parameter like weight or something. And suddenly we're like, wait, maybe this basic thing can be used as a biomarker if you're specifically looking at it according to the right context. And it is difficult to interpret like PCA plots and things like that. But there's definitely, I think, more power to those. And I think, yeah, you're definitely right on like the statisticians being like, you can't really do this for all of those columns. There's too many columns and not enough rows in this spreadsheet. And, you get into the problem of, I guess, multiple comparisons. You're like, well, we're going to do like 1,000 ANOVAs.
Yeah.
Multiple comparisons be damned.
Yeah.
And then you do your Bonferroni or whatever it is, and you're like, wow, nothing was important. Nothing changed. And I guess that's a whole separate conversation where I feel like sometimes sometimes I feel like the statistics gets in the way, or not, that's not what I mean. No, sometimes I feel like p-values get in the way, where you're all kind of rallying around this threshold, but sometimes I just use your eyeballs. Just like, look, these animals and these animals they are different. Maybe we weren't totally powered in this study, but like something's going on here. I don't care if the p-value was like 0.06 or whatever it was, 0.11. There's something to this. Maybe those are some of those more eureka-y moments to figure something out. But on that subject of it just sounds like we're at a lack of understanding how biomarkers, what is a biomarker, how to characterize it. And some companies, probably the really well-funded ones, will put work into that. Who's supposed to be, who does fund this? Who are the groups that are doing this? I guess you can go to NSF, NIH. How do we, where do we generate more funds for that? Or I don't know.
Yeah, certainly the big, NIH and NSF are, and probably USDA too, on somewhat on the maybe human and livestock side. But I'm guessing largely it's NIH and NSF where, NSF, it's got to be, I've never, I guess, really been funded with NSF, but it's got to be more comparative and not really with a clinical outcome or something, but it's if you can be somewhat comparative or more on the basic science side, it certainly can be fundable. And NIH certainly, I think a lot of people are probably looking at, biomarkers for some disease outcome or some outcome of importance, that but in industry, I think there's probably some of that as well, where if you think about the human or even in pet side, if there's a functional outcome that you can measure something, if there's a new metabolite, and some of, I know on the pet food side, and there's not a lot as much published as I think there probably should be, and based on how much work is being done. I know there's a lot of work being done, but you don't see a lot out there. But there's a lot of data being generated, I think, and I think it's that probably some of that data mining or even that confusion of what do we do with the data, because it is complicated. But I think there are some that are nervous about publishing that, because your point is well taken on. It's so the going through publications now, the first, I don't know, maybe a decade ago or whatever, maybe it's longer than that, but when these kind of quote unquote online journals came out and there's more open access, where there still might be a length restriction. You don't want to just write a novel that goes on and on and on. You want to get to your point, but the use of supplementary figures and tables, I think you're right. Some of that, probably the most important information in some of these data sets is in some supplementary table that some data miner will find later on. The authors, if you're in this, you're trying to interpret it, but you just can't see it. And like you said, I think it's very important to if some people don't want to share it, but most of our work, we share the raw data, we put into a database, or even if it's the means of, that, like you said, you might have a figure, but that the actual data are important as well. It might not, and it's, you don't want to be duplicating everything, but it is important that you have that there. And I think that's where in the future there'll be people finding this, so I think there's a lot of work being done. But yeah, it's going to be I think moving forward, is it going to be a collective effort of people looking at, I always kind of say the truth is somewhere in the middle. We're all looking at it from a different perspective, trying to see what does this mean, and what is real, what's going on here. And I think eventually, having clinicians look at it from a clinical perspective, like more basic biologists or microbiologists looking at it, all this stuff is being put into the I don't know, the scientific world, I guess, and hopefully at some point we can start, connecting the dots a little bit better. But I wanted to mention though something too. Yeah, looking at ratios. That's where when we start thinking about, we often don't think about it ratios of these different, but it could be even microbes where, if you think about dysbiosis, once you shift. Sometimes you look at several conditions, I think it's very difficult because there's not really a threshold. These metabolites, microbes, rarely, other than, I think, C. diff infection or something like that, you might say, okay, yes, this is a problem. But in many cases, it's like this slight shift. And at what point do you get this imbalance where now it's a problem, where these microbes have been there, or these metabolites have been there for years, maybe in an animal or a person, and it hasn't been a problem, but now it is. And I think it's maybe those ratios that we just don't understand probably, but and the thresholds, because people will say, whoa, fecal ammonia or some hydrogen sulfide, disulfide, or something like that. Or what's the problem? But no one really has a level that says once you hit a certain level, now that's a bad thing. We just don't know. It's kind of the ratios or the imbalance maybe that's there, and it's really hard to define that. And I think hopefully we'll have better. I guess that kind of goes for something like you think about blood cholesterol too. It's not just, everyone has cholesterol in their bloodstream in their body. It's just at a certain level now. That's more of a problem because it's a higher risk. I think it's the same thing here where that's a problem. And another thing too that I just wanted to say, when you get to the statistics and prediction, I think you're right that I'm never too. I understand that we need to have statistics. So it's there's something there, but it is difficult. And sometimes you get into, if you get too much into a set in a box and just say it's all about p-values and anything over 0.05 doesn't matter, I think then you've thrown out all this potentially important data that if you're just severely underpowered, like you even said 0.11, a lot of people would say, oh, you can't say that in a publication. But if you set up a study and it's still around 0.1, I still think there's something going on. It doesn't, you can't throw that away. At least maybe you can't say certain things in the publication, but as a scientist, I would say, well, I'm going to go back and maybe you power it differently, or you look at it a different way, or, at least if I was a manufacturer, you don't throw that away. You want to maybe go back and strengthen the evidence there and understand it a little bit better. But I think that's an important thing that some people are, and it's always like that reviewer number two that will just say, nope, it's not, you can't say anything about it. And I think, if the p-value is not quite there, I understand you can't be so, strong in your statements and everything because it is maybe a trend or something. But I think to not be allowed to say that there's something going on here is maybe wrong as well. But one last thing, I was writing a couple of notes here while you were saying things. We've used in the past, you have the PICO-A plots or PCA plots. So it's nice to see the big picture, but then if you can drill down, we've used things like random forest analysis to show which metabolites or which microbes are really have the largest impact on the shift. And it doesn't mean they're the most important, but it does kind of start pinpointing, okay, these are the ones that are really distinguishing this group and this group from each other. And it maybe doesn't always end up being that this is your biomarker, but certainly I wouldn't just, we often in our analysis, we've done the top 20 or the top 30, metabolites or something that seem to be moving the most, and whether it's diet or over time with weight loss or whatever. And it is different. It is interesting that I think that can be useful too, that if you can identify why they're changing or what are they a marker of, or why is this biomarker seem to be there? Sometimes it seems pretty easy, but other times we just don't know enough about it, and it's not easy. It's not simple. But I think some of that kind of taken from big data, getting to some, again, getting a hypothesis generated from the big data, that's, then you can go down another path. I think that's a nice part of the omics strategy. From an industry perspective, and well, even government, I guess too, we want all our tax dollars going to something useful, but that's the risk, right? You can, like you said, you can sink millions and millions into that side of it, of searching for some great thing, and you might not ever find it, or at least with the budget you have. So you have to do it strategically, I think, and so you have to be careful before you invest in, a huge project. You have to, I think, use screening tools, or maybe you have a sure thing and try it there first, and then go there. But it's kind of a balancing act, I think, of what your budget allows and what your what your goals are for that year or for that product, or whatever. You really have to lay that out. And I'm on the, I guess I'm the user, I guess, or the researcher on many of those, when I'm writing a budget. But I'll be honest with sponsors, this is your budget cap, but how best to use the money, or this just isn't feasible, or I wouldn't do it that way. I wouldn't waste your money on this, or use your money. Maybe waste is not the right thing, but you really have to know what you're measuring and what that means. So, and not that I always know what it means, but kind of just being honest with people, because you can get, I'm a believer, I think all these products, there's a lot of potential here for improving gut health and just host health in general, but you have to be real as well. Like, have the, kind of the real world, that these are the facts, that you're not just going to throw something in and success happens. It's you really have to have a lot of thought behind it. So unfortunately, sometimes with startups, or you have a new product, you don't know where to start. So hopefully, I can try to convince people to do a dose response study, and we'll measure our common targets. We don't need to get too heavy in the omics right away to see if we can have a good biomarker or an outcome that shows a benefit, and then you can go from there if you want to. But you really have to be careful what you pick there. And some things, if you're looking at fecal characteristics and things, it doesn't cost much. But you can demonstrate a benefit without sinking in $1 million in some kind of huge, I want to do metabolomics, proteomics, microbiome, all these things, when it start simple and at least keep it simple at some level, and get innovative on one side. If you want to differentiate yourself from someone else, that's fine, but you have to keep that in mind, I think.
That's a great point. And so a lot of it comes back. So what you said earlier about structure function claims, you're not talking about diseased or disorderly states, right? We're usually talking about a healthy population and feeding them something that at worst is like inert and at best is pretty healthy. And so how do you make a healthy subject healthier? And what is problematic is in animal research, it's not a problem actually. You have the 3 R's. You want to reduce the amount of animals you're using. What that means though is that when you detect an effect in your stats, it's a big effect. Like you need a big sample size to detect small changes, and a lot of what's happening in nutrition may be subtle, which is kind of why I feel like you could raise that p-value to 0.1 or even higher, because you're only detecting like the big hit you on the side of the head changes. And some people are kind of worried about like p-hacking and, oh, you changed your model to get significance, and I don't know, like whatever. There's an N of 6, so if you see something there, it's probably legit. Of course, and there's an exception to every rule on that. And when you get to the point of running a clinical trial, for example, it's really easy to get seduced by the omics. Let's just collect some more blood, collect some more fecal samples, run it through this or that. And it comes back to the definition of all these biotics, pre, post, syn, any of those, is that you have to confer a health benefit on the host. And whether, I don't know if it's been taken kindly or not, but, I have told groups like, don't, as you said, don't waste your money. Don't unnecessarily spend your money on a bunch of omic work, because that will not get you to that statement where you can say it confers a health benefit. I would rather if someone's got 50,000 or 100,000 laying around, first, congrats for just having extra money laying around. But up your sample size or up your power to try to be able to have as much of an opportunity to detect the right effect, that actual health benefit. And I guess I'm saying that makes sense when you're at the point of like, I know what my primary outcomes are. I know that this thing affects weight or, cholesterol or whatever it is. When you get to that level, I feel like you should put all your eggs in that basket. Of course, when you're doing all the production, in vitro, preclinical stuff up to that, it does make sense to cast a really wide net to try and see what's going on. And then the other thing is, as you were mentioning, so we've been talking with a psychology professor on the topic of high dimensional data sets. So you've got lots of columns, and you don't always know what they are. And in both psychology and in nutrition, you're measuring things that are kind of like immeasurable. I mean, health, you can't weigh health. You can't put it on a scale. And he gave an example. So for them, we're kind of coming at it from two different perspectives. So they might want to measure something like sadness or anger or depression. Can't weigh that. There's no colorimetric kit for that. You got to ask people or observe their behavior. And so there's a lot of questionnaires. And what's great about questionnaires is like, we know exactly what they're saying. Are you sad? More complex ways of saying it. But, they'll get at that with a lot of different questions. And then, they have this latent variable, sadness, and they have all of these known variables. They're known because like we wrote out what the question was. And then they do factor analysis, because the truth, as you said, is somewhere in the middle. It's a combination of a lot of these variables that is going to correlate with this unmeasurable latent variable sadness. It's the same thing like when you get an omic data set. They're not always labeled. It's like X1 through 100. You don't even know what that variable is. You might know it's bifidobacterium, whatever, but then you like go on PubMed and you're like, no one researches this. Yeah. So we have the opposite problem where we don't know what the questions are in our questionnaire, but we are trying to reduce the dimensionality of our data sets. Can we put multiple of these things together? A simple way is doing a ratio. More complex is doing a PCA, LDA, or any of those things. And I think that's kind of where this biomarker stuff comes in, where it actually might require all of those variables together to be able to adequately characterize how that population changed. It doesn't have to just be like microbiome sequencing or metabolomics. You can do those same approaches on the really basic clinical stuff. Like, I feel like you could combine fecal characteristics, just like observational even, softness, water content, short chains. You can put a lot of your data in a blender, I guess. Maybe not in a blender, because you hope what comes out is probably not just a slurry. But to look at these things, and I think I would hope that makes some of this a little bit easier. Easier is not the right word to use. But the thing is, like, when you get to the end, when you get that PCA, I feel like, okay, that's the beginning. Actually, that's not just the end, right? So you can separate your groups out, right? But now you have PC1, PC2, whatever they are. Those actually, you can do your stats on that new variable. That new variable can be correlated with other things, and so there's so much opportunity to figure out what's going on. I do, bringing it back to postbiotics for a second, though. This is bringing it like super back. There's a lot of components in them, and one postbiotic could be processed multiple different ways. The question is, do you think postbiotic is a temporary term? Like, if you see a benefit, you may then go down the path to figure out how it works, which is why we're talking about biomarkers and things like that. Mm-hmm. But say it is just a component like the cell wall or something, some oligosaccharide component. Do you foresee eventually just saying, all right, well, forget about the rest. We just want to purify this one thing because that's responsible for this effect? Or do you think there is something kind of unique inherent to this complex multi-component thing?
Yeah, I guess I think it probably could be both. I don't think it'll go away. I do think the nice thing with the postbiotic, there's, from a stability or viability perspective, that there's one of the worries with probiotic, eventually the viability goes down, where here it should be in most preparations more stable, and also the number of mechanisms you can hit, and not every product is going to hit every mechanism. Just like every probiotic, people have sometimes you'll see press releases or a story will go that, oh, probiotics don't do anything, or they don't do what they're supposed to do. Well, they're all tested for a specific function, typically like a probiotic or a prebiotic. The target was GI health, so don't say it changes my mood if that one in particular affects GI health, and that's the focus, barrier function or something like that. So that's one thing that sometimes we have to remember. Usually really from a claim perspective, you should be claiming what the data tells you. Not that it does everything. It does, might do A, B, or C, but not all of it, or it doesn't do X, Y, and Z. It does this. But people, I think, especially I think consumers probably just say, well, probiotics should do all of it. They don't know the specificity there. But and I think postbiotic, it gives I think if there was something really, really interesting, like on the heart disease side, TMAO or something, like there have been metabolites in the past that have come up and showed, oh, this is really, really important. And now, that buzz has kind of gone away. So I'm not sure how important it is, but some of them might be singled out, and, butyrate singled out a lot, for its effect, when in fact, I think, and people will do this with foods where they say, let's isolate everything and put it into a supplement, or, where usually it's still, we try to say, well, eat the whole food, because there's a lot of things we don't understand. So I think, I still think that the postbiotics will be there, because I think there are some advantages that they have. But I'm sure there will be, if there is a isolate or a metabolite or something that can be, that's really shown to have, trigger a certain receptor or something. I'm sure like a, I don't want to call it a drug, but it could be more it might go beyond what a food would be, or a supplement. It might be something else. I'm sure if it hits the right target, or it has the right effect, that some of that will be taken out. So I'm sure that will certainly happen. But I think I think postbiotics will be around, though. I think there's so much room for innovation, and just having this combination that whether it's gut function, immunity, and these other potential targets, to have a benefit. It's hard to prove if you want to be a naysayer, well, what's, or you want to know what's doing that, it might be difficult to prove what it's doing. And even that's the difficulty with even with prebiotics in vivo. How do you can do in vitro fermentations, you can do other things, but proving that group, small group of microbiota, digested or, broke down this prebiotic and then did this other thing, like you said, there's a lot of pathways here. And especially if you're talking about mood, going from the gut to the brain, yes, you have the vagal nerve and there are certain things that have been shown there, but there's other mechanisms but there's a lot of different things that happen before you get to the brain, and having that then change someone's mood or cognition or whatever it might be. It's hard to prove that. And so that's kind of a challenge, but I think if you can have, again, some key outcomes that you can show this is beneficial, then it'll be there. But so I think it'll be around for a very long time. I think because there's so many opportunities there too that despite its complexity, the complexity can be used for an advantage though too. From a market share perspective or kind of IP protection, you might be able to protect some of that with how it's produced and stuff like that. So, but one thing you said before, I guess in this process, I wanted to say too that even sometimes I get disappointed, and students certainly do, that we don't see the effect we hope to see with the treatment. But a lot of these studies, especially in the dog and the cat, but most of it in people as well, even if you don't have treatment differences, I know the sponsor always wants a treatment difference and a benefit. A lot of this, what I call this kind of characterization, and it might be kind of a descriptive data set, but even that's useful to an extent where we just take a fecal sample of a dog, and fully characterize the metabolites that are there based on, this diet or that diet or whatever supplement. That's useful in itself. It might not be what the company or the sponsor would want to see, but just characterizing that, because we just published a paper actually on a metabolomics data set with weight loss in cats. And so one of the reviewers is like, well, all these, like the super pathways are what you'd expect. And so they were kind of, I guess they forced us to say what's most novel. And we were kind of saying everything's novel because no one's ever published a data set like this before, but we had to kind of specifically call out some of those metabolites. But like you could say, oh yes, I think, blood lipids will go down, or these metabolites will go down or go up. But when you have, in that data set, we had almost 300 different metabolites that were changing over time. You'd have been able to predict a handful of those, but a lot of them, you have no idea what lipid metabolite, like some lysolipid that's modified, or, I didn't even know lysolipids were even a thing until we got these data sets, a few years ago. So I think the same thing happens with with the microbes too, that, just the characterization and description of this is a healthy state in a dog or in a human, and eating this diet, you have to be kind of specific there, I suppose. But I think people still argue what's normal or what's healthy in the microbiome, and it all depends on who you are, what's been fed, the environment, exposure to antibiotics. And so there's going to be this, like people said, kind of a reference range, if you will, although we don't even have the reference range for humans or pets. But I think if we keep moving forward, we'll be able to characterize that better, I think. And so, and I think something we've talked about before too is it's the easiest. So it's easiest to get hung up on is the taxa. But I really think not that the taxa don't matter, but it's really what the microbes are doing and what they're producing and how they're interacting that's really way more important. It's more difficult to measure that. So oftentimes we'll just do 16S sequencing and look at who's there, but there's so much redundancy and overlap that, I think with some of this, there is concern, and there should be, with different microbes basically going extinct, with industrialization or, from a food side, we're highly processing foods, and we're not, we're eating more refined foods. And so some of these microbes are going away over time. And so I think we should be concerned, but I've also questioned other quote unquote experts in the area. And I kind of say, well, in this data set, you showed that certain taxa went away, but did the functional capacity, did that go away, or is it still retained? And I think that's a key thing to think about. And so I don't think wild populations of animals or, people living in, non-westernized societies are necessarily the target. They can be useful, but, it's they might carry a lot of pathogens and other, some of these gene sets we don't want in our body as well. So so I think we have to think about that, but just characterizing these different populations, I think, is very important from that side. Again, at that each study, each data set, we're probably not smart enough, or we don't have enough information to make sense of all of it. But I think, slowly we'll be, as you, like you said, if you go into PubMed and there's no one else has shown this metabolite, on one hand, it's really frustrating because what do you say in your discussion, or what, there's nothing there. On the other side, maybe you've just discovered something, you don't know what you've discovered, but you're presenting data that's really novel. But it's hard to discuss because there's nothing else out there. So I think it's pretty, that's an important part of it. One other thing you said though too, I wanted to just come back if I could on the kind of the non-responder responder phenomenon, and more people are getting into the personalized nutrition, and the microbiome is becoming a key part of that. I think host genetics certainly our general environment is important, but a lot of people are even starting to think if we even stratifying, if you have a microbiome study, or stratifying based on their baseline microbiome, that if you have maybe you already have a high bifidobacterium population to begin with, you can probably, in some cases, you probably say they're not going to be very effective because their bifidos are kind of maybe as high as they could be anyway, like relative abundance. They're not going to grab a bigger part of the gut than they already have. So I think kind of there's a lot of people looking at that side of it as well. You have to think about if you stratify your or even if you do a baseline microbiome and then you only work with a subset, the implications of your data then only probably apply to that subset. So you have to keep that in mind, I think, from an application of the data. But there's a lot of that work being done as well. And people not arguing, but discussing what is the best strategy. Should you stratify? Should you do it this way or that way? And, we think about sex and age and all these other things. Your baseline microbiome is probably in the future, I think, will be, at least for some studies, will be a key determinant that help you be stratified, or I guess classify you in some way, I think, because it's and I don't want to get into enterotypes. I think put people in just like 3 categories. It's too is too general. I think it is, it's like different shades of gray. I think we're all a little bit different and your profile is different, but still having some level of characterization, I think, is important, and because I think it will affect how research is. Well, it's already affecting how some research is being done, and I think in the future it'll have a bigger impact that way. But in some cases, our animal population, it is what it is. So we're going to use the animals we have, but in if you think about free living animals that they're client-owned pets or something, or with people, you see a much greater variation there just based on what they're exposed to and maybe their genetics or other factors. And I think then it probably is more meaningful in those studies. So, but yeah, it's all of this adds to more complexity. But I think there's going to be I don't want to say it's going to get worse before it gets better, but I think scientifically we're going to be in the trenches for a while, but the payoffs and the outcomes are much greater than they have been in the recent past, I think. And so that's the exciting side of it, I think. And like you said, if you can identify some of these, a biomarker or metabolite, well, then they might be very successful on their own. And now you don't say it's part of the postbiotic. You say, I have this metabolite that does this, and then you can you can focus on that. And again, people have done that with some of the HMOs, the milk oligosaccharides, where some of the key HMOs now are their own product, where they've instead of trying to say, here's the profile of 50 or 100 of them, here's the key ones that we think are effective, and they've shown that. And whether it's young infants, but I think on the pet side there's possibilities of some of those products being there as well, and serving either in a diet or maybe as a supplement, or something like a probiotic if an animal comes into the vet clinic and needs to go on antibiotics or something like that. Here's a supplement that might help them maintain gut microbe or just even gut stability, whether it's microbes or just stool quality, so they're not getting diarrhea and stuff like that. I think there's a lot of opportunities there.